We are going to look at meiosis. We're going to have an overview on the different stages of meiosis, and we're going to track the chromosomal number and how chromosomes are dividing to give us the daughter cells. Let's start at the very beginning. We start with one cell. We have the condensation of the chromosomes, we have the doubling of the genetic material, and so on. And now we are at prophase of meiosis 1. So prophase 1 notice that we will have the formation of the spindle and the attachment of the spindle fibers to the chromosomes, and now the chromosomes are going to move and align on the equatorial plate. Homologous chromosomes are aligned next to each other. This is metaphase 1. Now we have anaphase 1, where individual chromosomes that are part of the homologous pair will travel each to a pole of the cell. This is anaphase of meiosis 1. And then we have telophase of meiosis 1, where the cell divides into two daughter cells, and now the number of chromosomes is halved. We started with a parent cell with 4 chromosomes, that is 2n, and now we have two daughter cells, each having 2 chromosomes, that is n, or 1n. It's not over yet. This is the end of meiosis 1, but at the end of meiosis 1, we directly go into meiosis 2. So in meiosis 2, we will have metaphase 2, whereby the chromosomes in each daughter cell are going to be aligned at the equator of these cells. Notice how the equator of these cells is perpendicular to the equator of the previous cell. And now these cells, they're attached to the spindle fibers, and we will have anaphase 2, whereby the spindle fibers will pull on the individual sister chromatids and divide them. Notice how meiosis 2 is similar to mitosis. And then we have telophase 2, the decondensation of the chromosome. And we now have 4 daughter cells, each with 2 chromosomes. That's n chromosomes, half the number of chromosomes in the parent cell, which was 4 chromosomes. That's 2n. So this is meiosis. And the cells that are formed from meiosis are the sex cells, so the sperm and the ova. This is how they're formed. Of course, we have a decondensation of the genetic material, and this is how we end up with 4 daughter cells.